Okay, it's 2 after, so we're going to go ahead and get started. Good afternoon and welcome everyone to the NINDS Hill Biomarker webinar. My name is Rakonda Metley and I am the operations coordinator. We will start off with a presentation of slides followed by your questions and answers, so please hold them all until the end. There is a chat section below, so please submit your questions and answers in there and we will answer them at the end of the presentation. This presentation will be recorded and placed on our YouTube page in the next couple of days. So I'll be sending that link along with the presentation slides to everyone that has registered. I will now turn it over to our moderator for today, Dr. Ram Aruchachan.
Okay, thank you, Rakonda. So I'm going to share the screen. Are you seeing in the full presentation mode?
Yes, you're good to go.
Okay, great. Thank you. So good afternoon, everyone, and thank you for joining the HEAL Biomarker webinar. I am Ram Aruchandran, and who is the program director for the HEAL Biomarker program. This webinar will be focused on the RFA discovery biomarkers and biomarker signatures to facilitate clinical trials for pain therapeutics. So helping to end addiction long-term initiative, or the NIH HEAL initiative, is a trans-NIH effort to improve prevention and treatment strategies for opioid misuse and addiction, and to enhance pain management. HEAL has more than 31 distinct research programs, and these programs are led by most NIH institute centers and offices. The HEAL Biomarker program is led by the Division of Translational Research, National Institute of Neurological Disorders and Stroke. And this slide shows the team involved in the HEAL Biomarker program. So Dr. Pelimanda, who was previously the lead of the HEAL Biomarker program, now she is leading the neurotherapeutics program, but she is kind enough to function as the advisor to this program. And this program is ably supported by Dr. Stilju and Ms. Medley. The first couple of slides are introduction slides about biomarkers, just to bring all of us on the same page. So this slide shows the difference between the biomarker, clinical outcome assessment, surrogate marker, and the clinical endpoint. Biomarkers are not to be confused with clinical outcome assessment, which measures patient symptoms or conditions, or the surrogate marker, which is intended for being a substitute as a clinically meaningful endpoint, or the clinical endpoint, which measures how a patient feels, functions, or survives. But rather, the biomarker has distinct characteristics of objectively measuring and evaluating as an indicator of normal biological process, pathogenic process, or pharmacological response to a therapeutic agent. As this RFA specifically calls for biomarker discovery and development, the next slide shows the different categories of biomarkers and how these categories are defined by the best resources, which is being jointly put out by the FDA NIH work group. There are 7 categories of biomarkers, namely diagnostic, monitoring, predictive, prognostic, pharmacodynamic, response, safety, and susceptibility risk. Each of these categories of biomarkers is defined by the context of use. What is the context of use? The context of use is the manner of how the biomarker is used and the purpose of the biomarker. So diagnostic biomarker is mainly for patient selection, and monitoring biomarker is detect a change in the disease status or indicating or finding the safety and providing evidence for the exposure. The predictive biomarker identifies individuals based on effect from a specific intervention or exposure. Prognostic biomarker is to mainly stratify patients, and pharmacodynamic response biomarker is the efficacy biomarker which could predict a surrogate endpoint, and also it is the response biomarker showing a biological response related to an intervention or an exposure. Safety biomarker indicates the presence of an extended toxicity for any intervention, and the susceptibility risk biomarker indicates the potential for developing disease or sensitivity to an exposure. So the next slide shows how these different categories of biomarkers can be used in the drug development process to clinical practice decisions. In the interest of time, we are not going to focus on the biomarkers that are only being interest to this RFA. So first is the monitoring biomarker. So this is being specially used either in phase 2, phase 3 and clinical practice, where it is serially measure the status of a disease or medical condition, or for evidence of exposure to a medical product. And the pharmacodynamic a biomarker or the response pharmacodynamic and the response biomarker shows a biological response has occurred in an individual who has been exposed to a medical product. And also note that this RFA that we are not focusing on mainly on PKPD or the targeting which one mainly, but rather than the response biomarker, which mainly suited for the phase 2 and beyond. And the response prediction biomarker can measure serially for the assessment of status of the evidence of exposure or effect of a medical product, and can be used in phase 2 and clinical practice. So as now we have finished reviewing biomarker categories, the context of use and how and where in the drug development process and clinical practices these are being used, now we will focus on the aspects of the RFA. So the goal of the HEAL biomarker program is to develop objective biomarkers and endpoints to facilitate the discovery of new non-addictive treatments for pain. This includes all 4 stages of the biomarker development, starting from discovery, analytical validation, clinical validation, and qualification. So in the discovery stage, it's been used to identify the biomarker, establish its association with the outcome of interest, and develop detection methods, conduct initial clinical validation. In the analytical validation phase is where one can establish the performance characteristic of biomarker, detection method that is acceptable to the context of use, establish the technical protocol and process, and also perform clinical validation studies correlating with the concept of interest. And in the clinical validation stage, you can establish the association of the biomarker, clinical sensitivity, specificity, positive and negative predictive values, evaluate the overall utility in the clinical practice and the drug development. This could be done in the large scale prospective clinical studies also. So qualification is where you take it to the FDA review after you have done all these 3 stages. For the purpose of this RFA covers all these 3 stages: the discovery, analytical validation, and clinical validation, depending on the project, what stage it has been you are going to submit. So this RFA is specifically to develop validated response monitoring and prediction pain biosignatures that will facilitate phase 2 clinical trials for pain therapeutics. This is a U mechanism. It's a UG3, UH3 mechanism. So it's a cooperative agreement and it is 5-year grant. The first closing date is March 11th. And the second one is going to be June 23rd and October 13 and February 23rd in 2023. And you can see that in the RFA itself, and here we have put the review date as well as the start date. So already, as I have mentioned, this is the HEAL initiative. It's a trans NIH initiative. So more than 12 other institute or centers and offices have signed onto this RFA. So this is not specific for NANDS mission focused pain condition, and it is applied to all pain conditions. That's why this RFA is calling even for biosignatures, multimodal biosignatures for more than one pain condition. And as this mechanism is cooperative agreement, depending on the pain conditions of your application, one of the listed scientific contact of other institutes here, which has been shown as scientific contact, and this is being in the RFA also, they will serve as the subject matter expert once your grant is being awarded. So this slide shows again the mechanism. So it is a UG3 phase 1, which has an exploratory mechanism for 1 to 3 years. And the budget is limited to 500,000 direct cost per year. And then the UH3 phase is the phase 2, and it could go up to 1 to 3 years, and the budget is limited to 1.5 million direct cost per year. And the total project duration is 5 years. And you have to note that UH3 cannot exceed more than 3 years. And we intend to fund 2 to 3 into 2022 and 5 to 6 awards in 2023 and 3 to 4 awards in 2024. So this slide shows the go, no go milestones between UG3 and UH3 should be extremely clear, quantitative, and definite milestones. Administrative review will occur at the end of each year, and the transition occurs via the administrative review, successful achievement of annual and UG3 transition milestone, overall feasibility of the project achievement, and also it depends on HEAL programmatic priorities and availability of funds. So the milestones, the go, no go milestones must be met to transition from UG3 to UH3 phase. And the milestone could address, here are some examples. Evidence that study planning and team coordination is in place. Evidence that metrics for clinical enrollment have been met. Evidence that quality assurance parameters for sample collection or detection method performance have met. The desired magnitude and the reliability of the association between the biomarker and the target engagement or responses to an intervention in preliminary studies. And there should be the precision, accuracy, and dynamic range of the biomarker detection method can be also be a milestone. And the initial prediction accuracy of the biomarker signature for the response to the therapeutics. So next slide shows the application entry criteria. So here, a good application should have a pain condition or several pain condition that may have a common pathophysiology, a biomarker or a biomarker signature from several or possibly multimodal components, strong biological rationale, strong clinical rationale. So that means you have a robust data showing that the biological rationale and also data for showing the clinical feasibility. Preliminary data for the proposed therapeutic agent has a robust efficacy, a working hypothesis regarding context of use, and how biomarkers will be used in the clinical trial, a clear plan for rigorous standardization of sample collection and detection methodology, multiple principal investigators, team, organization, depending on what you are proposing in the application. And also the team should be knowledge and technical proficiency should be very much documented for the review. So as we are talking about the multi-component biomarker, we thought about spending 2 minutes here to understand the current FDA perspective of this multi-component biomarkers. And the FDA had a recently had a workshop, and this multi-component biomarker is taken out from the current perspective, is taken out of the recent FDA workshop on identification, concepts and terminology for multi-component biomarker. That was the workshop conducted recently, that on March 23rd and 24th. And that workshop was to mainly get clarity regarding related concept and terminology. So after getting the feedback, now FDA is generating a white paper before deciding on the terminology. But the current regulatory view of FDA is, in principle, that whatever applies to the single biomarker component, it will be applied to the multi-component biomarkers. That is the evidence of analytical clinical validation for the proposed context use. How does multi-component biomarker add value compared to a single component? Rationale for inclusion of the different component and for weighting. Importance of individual components to the whole. Interpretation where one or more components may drive the overall results. So please consider when you are assembling your application about this component biomarker or multimodal biomarker, but this is the current regulatory view of FDA. And so either you have to justify the context of use singly and also as a whole. And when components derived from machine learning, that consider that methods for future selection, and you need to provide justification. And also the small differences in the input data can dramatically input the output models. Consider how critical are these specific assays, and then you need to justify them. Characterize the link to causal pathway and the pathophysiology. So please keep in mind when you are putting the application that this will be also will be reviewed carefully. And so now that as we are talking about the context of use, so points to consider for generating the context of use when you write the application. So identity, aspect that is measured, and the form in which it is used for biological interpretation, species and characteristic of the animals or subject studies. I mean, like if you are doing early studies in the animal, that you should need to characterize and definitely define the species and how it should be relevant to the human studies also. Purpose of use in drug development circumstances for applying the biomarker, interpretation and decision action based on the biomarker, and your study design should match your context of use. So when you are actually writing your application, your study design should match the context of use, what you have been choosing for the biomarker. And this is even for the qualification program, this is what FDA expect where in the use of statement and the condition for qualifying use in the qualification program. So it is always better to keep this in mind when you are writing your application. So next, again, this slides again emphasize that this RFA is calling for response monitoring biomarker signatures. So definition, again, we want to reiterate here too, response monitoring biomarkers or signatures, which will indicate that the therapeutic agent has engaged with the intended biological target and can be followed over time. And the response prediction biomarker signature means that it will indicate the individual is likely or not likely to respond to the therapeutic agents. So how these biomarkers will be used in clinical trials, because this will be evaluated in the application too. So when you choose a biomarker, that response monitoring biomarker should be the early indicator of efficacy in phase 2 and even phase 1B trials. It could be an adjunct endpoint in early clinical trials. When used in combination with drug exposure, could be used to select dose levels in early clinical trials for acute or chronic drug studies. So these are the characteristics of how they will be used in the clinical trial for a response monitoring biomarker. So if you are choosing a response prediction biomarker, then your end result should be a stratifying patient subjects into more homogeneous group like to respond to a therapeutic mechanism of action. They could reduce the variability in clinical trial design. And for example, If the therapeutic mechanism depends on increased expression of a particular voltage gated channel, it will allow a more informed method for treatment arm assignments. Likely more helpful in treatment decisions, allowing more accurate choice of therapeutic mechanism for individual pain patient. So now we are going to see the examples of responsive applications. So a responsive application could be one that have identification and optimization of a single biomarker or a biosignature that can accurately monitor, indicate, or predict response to a specific therapeutic agent or therapeutic mechanism for a single pain condition or for several pain conditions that may have the same underlying pathophysiology. Or it could be a general therapeutic mechanism using a well-validated or approved pain medication, but it could be a biomarker probe with all samples from human sources. And it could be a novel therapeutic agent also that has but demonstrated a robust efficacy and using non-human samples as initial identification followed by a translation of the detection method in human samples. A non-pharmacological therapeutic regime could be also used. It's accepted, but it should be widely used in the treatment of pain. So a couple of other examples that you can establish a single set of detection method component also, and you can identification and optimization of the biomarker or biomarker signatures using centralized resources and optimizing the algorithm or a signature and translation of biomarker signatures developed in animal to detection method to human. So the translation from animal studies to human studies responsive to. So example of non-responsive applications, applications proposing to develop any biomarker signatures categories other than the pharmacodynamic response monitoring or response prediction biomarkers, and the natural history studies without biomarker identification, development of detection technology and early validation, biomarker identification or validation proposing only animal studies without confirming using human tissue resources, use of non-standardized sample source selection and large prospective design clinical validation studies, therapeutic only the therapeutic target validation studies and development without any biomarker involvement, and determination of efficacy and development of candidate therapeutics as the primary goal, and biomarker signature development not on addictive therapeutic option or for pain conditions. And then finally, the main application that do not include annual and UG3 to UH3 go, no go milestones. So as a general advice, so you need to these are the important points you need to cover in your application as the unmet need. So the proposal should be able to define what the current standards are and what performance metrics and methods of detection and how it is being improvement over those standards. And also in the study design, you need to pay attention about the biomarker signatures utility for decision making within the context of use proposed, and the statistical analysis plan should be clearly described and appropriate for the context of use, and also the identify and the address source of variability. And the study section, so this RFA will be reviewed in NANDS study section. So reviewers come from a variety of expertise and to evaluate different parts of the proposal, and they don't have, please don't assume that they know the state of the science within your field. So if your great innovative ideas to be turned into a good application and to be scored well, please read the RFA carefully, and more specifically the research strategy section and also the application review information, paying particular attention to what it says under the specific to this RFA, because this is the specific review criteria for this RFA, and reviewers will pay attention to this when they evaluate the application. And also don't hesitate to contact us if you have any question regarding this. So before taking any questions, I just want to highlight in this last slide about the funding opportunities available at NNDS biomarker program. So the development, analytical validation, and clinical validation PARs are available for biomarkers and biomarker signatures for neurological disorders, including pain conditions that are within the mission of NANDS. So the program director and the program contact for NANDS biomarker program is Dr. Carol Taylor Bird. So you can contact her, or if you want to contact regarding pain, you can contact myself also. So thank you. And now we would like to take your questions if you have any.
Okay, thanks, Ram. So for those that joined in later, please submit all of your questions in the chat feature below. But so far we have one question, and it's, does the preliminary study has to be our own, or can we gather from published literature?
It could be gathered from public literature too, but then there should be some evidence that your lab being able to follow on that study or have done some related studies to what you are taking from the publication. It's how it's being, the reviewers will take that.
Okay, that's all we have so far, but we'll give everyone time to get the, okay, they're coming in, okay. Thanks for the great presentation. Is the therapeutic response a required one, or applications proposing to describe biomarkers alone will be considered?
Biomarkers alone. So, sorry, I'm reading the question. So,
Oh,
so yeah, the biomarker alone will be considered, but then you need to actually there too, you need to explain how this will be correlated with for the therapeutic response. Marianne, would you like to take on that one too?
Yeah, just want to emphasize what Ram just said, that actually the therapeutic response is vital to this particular FOA. So remember that the biomarker type has to be either one that looks at monitoring response of a therapeutic, like a pharmacodynamic biomarker would, or it has to be one that will predict response to a therapeutic. So somewhere in there, a therapeutic has to be involved.
okay, next question. Will electrophysiologic biomarkers from humans be responsive?
Yes, they are responsive, but the answer for the previous questions actually applied for you too. So if you are using electrophysiologic biomarkers, and you need to define how this will be a therapeutic response or therapeutic monitoring biomarkers.
Next question. The study lead has to be in the US, right? Can we perform international multi-center studies with corporation partners in Europe?
Yes, it is allowed. You can do that.
Okay, next question. To clarify, therapeutics here are only drug therapeutics, or can we also consider future neuro, oh, sorry. Can you see that word?
Yeah,
Yeah,
neuromodulatory therapies.
right.
Yeah, that's a great question. Yeah, you are allowed. It's not only the drug therapeutics. You can use the neuromodulatory therapies too. But you are using word using future. That's what I don't understand whether it is already a standard practice or whether that's the therapy is going to be developed. So as a general point here, non-pharmacological interventions are also accepted here if you are going to bring biomarkers for that as a therapeutic and the monitoring ones, which is widely accepted in the pain field right now.
Yeah, thanks. I guess there's no FDA approved invasive neuromodulatory therapeutics at this point. So, but
That's fine.
yeah.
Yeah, if it is accepted widely in the pain field, right, currently, then that's okay.
Yeah, I guess it depends on your definition of widely accepted. In the neurosurgery field, for example, deep brain stimulation, off-label DBS is being performed regularly, but still off-label from an FDA standpoint.
Mhm. Yeah, I think
It doesn't have to be approved by the FDA in order to look at it.
Sorry, I missed the first part. It does or does not have to?
It does not.
Okay, thank you.
And next question, great presentation. It seems to me that there is some overlap among predictive, prognostic monitoring, and suspectably biomarkers. Can they be used interchangeably?
Well, that's what we said. It is going to be a predictive. Yes, there is a overlap, but not definitely. You cannot bring the prognostic and the susceptibility biomarkers into the predictive response and the monitoring response biomarkers. You need to have that kind of a clear definition for that.
And I'd like to just add something to what Ram said too. When you're looking at the FDA/FNIH's definitions of those biomarker categories, predictive always involves a therapeutic. So, and that's the difference between predictive and prognostic. Prognostic usually refers to predicting what's going to happen from a disease state, not necessarily from a therapeutic. And usually not actually. So predictive, think about, looking at the future for a therapeutics effect. Prognostic, looking at the future for a disease's effect. And monitoring can include both looking at the effects of an agent that's, been given to a patient, but it can also be looked at in terms of monitoring the status of a disease over time, such as tumor growth or something like that. And susceptibility is usually you're looking at a patient that doesn't have a disease, but you're looking at the risk of eventually having the disease.
All right. So if there are no more questions, then probably you can email to us if you have any questions. Please email to me. Then we will reply to you. Or if you need any meetings also, please email us, and then probably we will be able to schedule a meeting also.
Okay, thank you, everyone. And again, this presentation will, along with the slides, will be sent out in a couple of days. Hope you enjoy the rest of your week.
Okay, thank you everyone for joining.